from what I understand, you have to take the gene of an Indian elephant, which is the closest thing to a mammoth.
Yeah, let me walk through the whole
Please.
process.
Yeah.
So first you have to find ancient DNA, which is pretty shitty on a good day. So the minute we take DNA out of our bodies or out of anything, it starts to degrade at an insanely rapid rate. So we definitely need to find a lot of samples. So we actually have about 109 mammoth samples ranging from 3,000 years old to 1.2 million years old, which is awesome.
Wow.
But it's also fragmented. It's like a shitty jigsaw puzzle that you don't know what the box is, and someone's stolen part of the puzzle, and then, oh, by the way, people have taken other puzzle pieces and put them in there. So there's all kinds of problems with that. So this is really an AI and compute problem. It's not as much a human problem. So you have to get a lot of samples first, and then you have to start mapping them to their closest living relative. And genotyping allows us to understand that that's Asian elephants, right? So Asian elephants are 99.6% the same as mammoths. They're actually closer related to mammoths than they are to African elephants.
Really?
Yeah, which always blows people's mind. That and the fact that mammoths were alive when we were building the pyramids, or aliens or whoever was building the pyramids. Like literally, like humans were building the pyramids while mammoths existed. And sometimes that blows people's mind because they always think of them as in this like weird, like prehistoric, like 65 million years old dinosaur.
So if you have woolly mammoth and you have 99%, how do you bridge that gap? How do you create?
That's synthetic biology. So you never have to get to 100%, right? You need to get to probably
synthetic biology.
Synthetic biology. That's where you are using all of these different genetic tools. Probably heard of CRISPR, all these other things. Genetics, which is it breaks the DNA. It's not always the best tool. We can now actually make individual edits to, when you think of the DNA double helix, right, in those rungs of the ladder, those individuals are called nucleotides. We can change the letters. Like, that's how precise we can be. We can say at spot 4,000,008, I need to change that letter. And so you change that letter. And then other times you actually synthesize big blocks of DNA. So when you notice that in the mammoth and in the Asian elephant, there's a difference, and if it's in these certain like protein coding regions, in all these different regions of the genome that drive phenotypes or physical like attributes, like the curved tusk, dome cranium, small ears, the subcutaneous fat layer, and then hair and coat color, you can actually then engineer that into the Asian elephant, right? Because you're only really looking at that 0.4% difference, right? It's still a lot of numbers. But you're only looking at that, and so the better you can be at software, and the better you can be using AI and computer models, the less edits you have to make, right? Because you're really just trying to target